Okay, hi everyone. We are live. Thank you so much for joining us again today for this biotechnology live stream. For those of you who have been here before, you know who I am, but I am Dr. Danielle Snowflack. I am the Senior Director of Education at Edvotek, and I'm happy to be back with you again today to bring you this biotechnology training in the comfort of your own home. For those of, whoops, went too far. For those of you who are unfamiliar with Edvotek, we are the biotechnology education company. We were founded over 30 years ago by Dr. Jack Cherkian, who was a professor of biochemistry at Georgetown University. And this was a super exciting time for biotechnology and molecular biology. And there were so many exciting developments going on in the research lab that weren't being brought into the teaching lab. And so that was the genesis of Edvotek. And so since then, we have worked with educators all over the world to help demystify science and to foster the next generation of scientists and science enthusiasts, people who are excited about science and love science. And we're doing this through hands-on active learning activities. And so we hope to make biotechnology accessible to all labs, whether they're research labs, outreach labs, biohacker spaces, or educators. You know, we just want everyone to be able to try these techniques, try the biotechnology, and really see how it inspires them. And so the demonstration we're doing today is being recorded. And the slides will be available on our website. So if you want to be notified when they're posted, Maria has now posted the link in our chat window. And so I will be able to send you the materials after. And we're also offering professional development certificates for those who are watching. If you want us to send the certificate, please indicate that in the form. And so the link will only be live until around 5 o'clock Eastern time. So please be sure to complete it. And we'll send that to you in the next week. So let us get started. So today we're talking about quality control and how Edvotek ensures that all of our kits are ready to go out of our lab and into your lab. And this is a really important part of what we do because we want to make sure that the experiences that you have with your students go as well as possible, you know, from opening the kit to performing it to analyzing the results, right? Because, you know, labs are hard and science is tricky and messy, and we want to make it as easy for you as possible. And so we have a quality control protocol in place. And so the definition of quality control is a system of maintaining standards in manufactured products by testing a sample of the output against the specification. So basically what this means is that when we make our kits, we test them before they leave the facility to make sure that they give us the results that we're expecting. And so not only do we test them, the materials, when we receive them at our lab to make sure that they are up to quality, you know, we test them after we make the kit, and then we test it at several times, you know, a year after the kit is made, to make sure that the kits and the experiments are reproducible. We continue to test the kits from time to time to make sure they continue to work, even after time spent in our warehouse, in shipping, or in your lab. And these steps ensure that you're going to get the best possible results with our experiments in your lab. So today I am going to walk you through our QC process while actually performing one of our experiments. And so we're going to go through this process with an electrophoresis experiment. Electrophoresis is a biotechnology technique that is essential to the biotech lab. It's used in labs across the world every day to help us analyze, you know, DNA, RNA, and other materials. And so electrophoresis uses an electrical current in a porous gel matrix to separate molecules by size, shape, or charge. And so with a lot of these dyes, we're going to be talking about charge and how they move within the field. And these are based on the physical properties. And this is based on the physical properties of the molecule. While it's very powerful, it's easy to do in the classroom lab. It's easy to do in the context of outreach spaces, in museums, or even in your basement, like I am doing today. And so the kit we're running is S46, which is linking STEM to Agarose gel electrophoresis. This kit is going to separate brightly colored dyes into separate bands. And I'm going to run you through the process of testing the kit. I'm going to talk about how we develop the kits, what we're thinking about in the lab, because we want to make sure that we make experiments that fit into your curriculum, that cover current events in biotechnology, and also that your students will like and that'll be reproducible. Furthermore, if anything goes wrong with your experiments, because mistakes do happen, we're going to tell you what information to collect so that we can help you figure out what went wrong and how you can fix it. So let's start with equipment. So the vast majority of the equipment that Edvotek, distributes to our teachers, it is designed by us with teachers in mind. And so we work with teachers and we work with industrial designers to create this equipment. We want to make sure it's durable enough for the research lab, but easy and intuitive enough for students to use without a problem. Because, we know that things can be hard, like running these experiments with a room full of kids who might not have any experience. We want to color code things and make them easy to open and easy to see, and easy and fun to use. And so, when we go into thinking about a new piece of equipment, this is what we're thinking of. And so this means we go through several iterations of designs, testing prototypes in our lab before settling on a final design. And after that, we will get prototypes. We will stress test all of the equipment in our own lab before we make it available to teachers. And furthermore, those stress tests include usage in our labs every day. So we are developing our experiments with the same equipment that we are using with you in workshops, that you're able to get into your classroom, because we really want to make sure all of the systems work well together. All of our assays are developed using our equipment that you'll be using in the lab so that we know our results are reproducible. Furthermore, all of our equipment is going to come with an industry-leading warranty. This basically means that if something goes wrong, we're gonna make it right and we're gonna help you fix it. And even if the equipment is out of warranty, definitely give us a call because many things can still be repaired. So today I'm going to use a piece of equipment that we designed at Edvotek. We are going to be separating dye molecules using a horizontal gel electrophoresis chamber. The unit today I'm using today is the M12. So that is going to be the unit on the left side of the screen. You've seen me use it in workshops before. It is a great unit for running electrophoresis. And so we actually designed this unit a few years ago with many features to make electrophoresis easier in the classroom. We had a previous M12, people loved it, but there were things they wanted us to do better. And so we met with industrial designers, we met with teachers, and we redesigned the equipment. And so, for example, some of the features that teachers love is that you can run 2 student groups in one unit. Let me actually bring up my webcam so that you can see it. We have that feature. The unit, the trays will only fit into this chamber in one direction. The lid only fits on in one direction. This is going to make it harder for your students to accidentally reverse the current during electrophoresis. And when you're done, we have these big buttons that make the lids easier to open. So that was that's a common complaint with electrophoresis equipment, is sometimes the lids are hard to open. So we put these push buttons on to make it easier to open. And then we have a pour spout here in our electrophoresis chamber, which is going to make it easy to get the buffer out. And so these are just some of the things that we think about when we are designing equipment for educators. How are we using this in the lab and what can we do to make it easier? And so we actually have an all-in-one solution that we've recently designed, the Edvotek Edge. And this unit combines the electrophoresis chamber, the power supply, and the visualization system all into one easy-to-use unit. And so we designed this unit taking some of the best features of the N12, and we used it to influence our design. And if you want to see how we designed the M12, what you can do is actually go to our blog, which is blog.edvotek.com, and search for the Edge, because we have pictures of the prototypes, the development work we did in-house, and a lot of the behind-the-scenes work that you don't think of that goes into our equipment. And then after we build the equipment, we test it before we send it out to you to make sure that it works. So that's one set of tools that we develop for our teachers and how we test it. And so we're going to use this equipment to perform our experiment today. So each kit and each experiment is going to go through 3 main steps before it leaves our facility so that we can ensure that it is working for you. And so this is our QC process. And, this is going to be like that flow chart where we saw those 3 arrows. We go pre-QC, we're going to go production, post-QC, and then our long-term testing. And we do this for every experiment because we want to ensure the best results. And so first we start by testing our raw materials. And so this is important not only in, our biotech lab, but also it's going to be important in pharmaceutical manufacturing, because you want to make sure that your raw materials are working. And so this is going to include our DNA for our DNA electrophoresis kits, the agarose, the dyes, even the plastic tubes that we include with the kits. We want to make sure that everything is the highest quality for our teachers. And so some of the stuff we make in-house. So the DNA in our DNA electrophoresis experiments, the vast majority is, grown up in cells and made and sent to you, and made completely in our house. Other stuff, and so other stuff we're going to purchase, like the agarose, but we still want to make sure that the agarose from one kit works the same as the agarose in the next kit and the next lot of the kit, so that we can ensure that you'll get the same results regardless of when you perform the experiment from year to year. And so, we analyze these reagents to make sure that they work with the experiment and that they're consistent. We assign each reagent in-house a lot number so that we can track it in our system and we know which kits a specific reagent goes into. And this is our pre-production quality control process. Basically, we want to make sure the raw materials in our kits work before we build our kits and send them to you. The raw materials, then they're going to move to our production team. And you can see here a picture of one of our production team members. And in our production team, the team is going to aliquot the materials and assemble them into kits. For example, this is going to be dispensing electrophoresis buffer into bottles, aliquoting dye samples into our quick strips, and then putting all of these pieces into the box and making sure everything's in the box. Once assembled, a kit is selected at random from the assembled kits and sent to our quality control team for analysis. And this is our post-quality control, our post-production quality control analysis. And I'm going to run through that entire procedure with you now. So let's go back to our camera so that I can run through this with you. I'm going to put my gloves on. So this is a brand new kit. I haven't even opened it myself yet. So we're going to run through this process together. Let me get this out of the way. I have a copy of our QC sheet. This contains all of the components, their in-house names, the lot numbers, and the volumes that are supposed to be dispensed. So basically we keep track of everything that's supposed to go into the box. And we start with a visual inspection of our kit. So here's the kit as you would get it. We just changed to this new packaging, which I think is really pretty. And so we open this box and we start with the question, do the samples match the description? And so I'm going to start with the quick strips. Here we have our S46 quick strips. These quick strips are going to contain our dye samples pre-aliquoted for use. And so, let me open these up. One thing you can see is that the storage instructions are here right on the bag for the reagent. So you can see this says storage, shows a refrigerator, and it says 2 to 8 degrees centigrade refrigerator. And so we're letting you know the storage right there. I'm going to open this up. And so these are our samples for electrophoresis. Now electrophoresis is a very versatile technique. It can be used to separate a variety of molecules, nucleic acid, DNA, RNA, and proteins. And in this experiment, we are going to, we perform electrophoresis of these solutions of brightly colored dyes. The samples are in the quick strip format, individually aliquoted by us to minimize the pre-lab prep on your end. And so in the visual inspection, we're going to look at the strips. So we look here, we say, is the foil tightly adhered here to the plate? Do we see any rips or tears? Is it properly labeled with a proper lot number? And then we would record the lot number on our sheet. And so we're going to need 30. So when we run this experiment, we need 35 microliters per sample. And so what we're going to do is I'm going to look at the, one more thing to look at. You know, we have 45 microliters per well, and it's going to be in 10 columns. And so what we can see is that we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 columns of samples. They're all full. So basically that's going to indicate that our samples were properly dispensed. I am going to pull off one of these strips, and then I am going to measure the volume that is in one of these wells. And so, so basically we need 35 microliters per experiment. As you can see here, we actually put in, can you see that okay? So we actually put in 45 microliters per well. You know, we want to give you extra in case there's a problem, in case your students are pipetting inaccurately. And we also build it enough because we do have basically our measurements are plus or minus 10%. And so what does that mean? That basically means that, you know, we're going to dispense 45 microliters, but we accept a pass as anything that is either above or below 10% of that number, which is 44.5 microliters. So that's so anywhere from just over 40 microliters to just under 50 microliters. Again, you're always going to have extra sample just in case there are pipetting errors. But you know, we give you extra, but we want to make sure that it's there, and we want to make sure that it's accurate. So I have my pipette set for 45 microliters. And you can see we have 45 microliters in this sample. I actually left a little bit behind. So it's probably right about at 45 microliters. Okay, so you can see the tip is full. You know, my thumb is off the pipetter. So I've pulled up 45 microliters. I'm going to just put that back into this tube for now. And what I would do is take this form and mark 45 microliters. And so what we're going to do is we actually keep track of all this information in-house, so that we know where we're starting when all of the kits go out. So if you tell us, you know, I have something from this lot and it's gone down to 20 microliters, you know, we're going to go back and test all of the other, look back at the other kits to make sure that we don't have any problems. So we would go through all of the strips in one of the, check the measurements of all of the strips in here to make sure that all of that matches up. So next question, you know, is the volume or the mass of something, of a component, going to be correct? And so let's look at another component. So here we have our electrophoresis reagent bag. And so we're using Agarose. Agarose is the matrix for our DNA separation. And we can think of it like a scientific jello. The agarose is going to act like a strainer or a sieve that helps us separate molecules by size. And we actually provide it as a powder. And so, you know, I don't have a balance that is accurate enough here with me. But if I did have that balance at home, what we would do is measure this and we would record the mass to ensure that it is 3 grams or more so that you can do your experiment right. The next thing to talk about is are all the tubes tightly sealed? So you could imagine in shipping, if something is not tightly sealed, it could spill, you know, and then you wouldn't get the amount of buffer that you need. Or you could imagine if something is not tightly sealed, you'll get a lot of evaporation. And so this is our electrophoresis buffer. It's provided as a concentrate, and it's a mixture of buffer and salts. And so what we can see is that, you know, it was tightly sealed. You know, the bottle is not leaking, and so that's good. You know, that means that this reagent is ready to go out. And so electrophoresis buffer is an important component of these experiments. It's a mixture of buffer and salts. And there are 2 reasons we're going to use this in our experiment instead of water. Since electrophoresis uses electricity to separate the molecules, we need to add some ions to help the electricity move through the gel. We also want to, since we're working with charged samples, we want to keep them at a pH that does not disrupt their charge or structure. And so this buffer comes to you as a 50x concentrate, which you dilute in your classroom. And we have a great video on dilutions if you want to learn more. And we also have a video that talks about what happens if the buffer is not prepared right, because that's a place where the experiment can go wrong, and that will show up in your results. The next step is going to be to use the reagents from the kit to perform the experiment. So I'm going to be honest, I'm going to cheat a little bit here because I made the gel and everything ahead of time in the interest of time so that I could run through the experiment, because it does take a little while to prepare the gel. So I've got my electrophoresis chamber here. And we're going to do the experiment using all of the reagents in the kit. I'm going to take our gel, I'm going to place our gel in the M12. So I've prepared this gel already. Again, we have videos on how to cast gels if you have not done it before. But basically, the gel casting system is kind of like a Jello mold, a scientific Jello mold. And we use it to create the shape of our gel. The wells are formed by the combs, and that's where we're going to place our samples. And I am going to place this gel in our chamber. And so basically we have the instructions handy, and we're going to follow the instructions. I already made the gel following this protocol. And then we are going to run the samples following the protocol, exactly like the students or like will be performing the experiment, to ensure that when you do the experiment, you get the same results we do. So let me get this out of the way. I am going to use the M12. Let me cover it. I have electrophoresis buffer here that I've prepared. Again, you know, in the interest of time, I pre-prepared this and I have it ready to go. But we generally try to use the stuff from the same lot so that we're testing the entire experiment together, all of the components together. I'm using the M12, and I'm actually going to test 2 samples in this gel. I have 2 sets of wells because I want to check for the consistency of the samples from strip to strip, right? Because not only do we want to make sure all of the reagents work from kit to kit, but we also want to make sure the results from kit to kit are consistent. So what I am going to do is I am going to use my adjustable volume micropipette. I'm going to set it to, if you see here, we say 35 to 38 microliters. I'm going to load 35 microliters into each well. Then I'm going to be using an adjustable volume micropipette. Which is a tool that we can use to make precision measurements of very small volumes. And so for the experiment today, we don't necessarily need to make the most accurate measurements. So if we were in the classroom, you know, we could use transfer pipettes to do this, and I have loaded the gels and run them using transfer pipettes. And it works. It actually works really well for students. You know, if you don't have the pipettes, you know, it's an affordable way to do it. But because this is our quality control protocol and that we're following, what I want to do is I want to make sure that the exact same amount is loaded in each of these wells. And so even though the samples are concentrated enough that if I didn't get the same amount in each well, you know, we would still get good results. You know, I want to make sure that our kit is consistent from gel to gel so that, you know, we can ensure that the kit is working. And so again, the adjustable volume micropipette allows me to make precise, repeatable measurements for our quality control experiment, right? So we want to make sure, so I'm going to just keep loading this gel. Oh, I made a mistake there. So that could affect my results. We'll see if that happens. I kind of missed the well a little bit there. It is not the easiest to talk in pipette, but you know, when we're doing science, you know, we are humans. Science is a human endeavor. And it's important to show that there are mistakes and science can be messy, you know, because these are learning experiences in the lab. And so, you know, students who are performing experiments, that you are doing in the class, it's important for students to not only discuss what goes right with their experiments, but also goes wrong with their experiments. Because even when you get negative data, it can give you information about the next steps to take. Our hypotheses are not always proven correct, and that's important for students to realize. You'll notice I am changing the pipette tip between samples. This is going to prevent cross-contamination between our samples. Again, because we are quality control testing this, we want to make sure that each sample faithfully reproduces the results that it should produce. And so we don't want to cross-contaminate. Now, again, for these dye samples, if you're in the classroom and a student accidentally, uses the same pipette tip for two samples, is it going to be the end of the world? Is it going to ruin this experiment? No. But again, because we are being extra careful, because this is our quality control testing, we want to make sure to use the best possible biotechnology technique when we're working with our stuff. All right, so I've got these two gels loaded. We're doing the experiment in duplicate, so A and B. We're going to put the lid on our chamber. Oh, one thing you'll notice is that the samples do sink into the wells. This is because our dye mixtures contain buffer and glycerol. If the dye was only dissolved in buffer, it would immediately mix with the buffer in the buffer chamber. And so if we did see that when we were loading the samples, if we didn't see them sink into the wells nicely, that would be a quality control concern. Did something happen in our sample prep so that it didn't work right? And then we would take that information and we would go back and analyze our raw materials. So we can see that our samples were more dense. They sank into the wells nicely. I've put the safety cover on our electrophoresis chamber, and I am now going to turn on the current. So I have set the current, I'm using the new M12. I've set the current for 150 volts. And so that should take us between 20 and 30 minutes. And so we are going to stay within that time frame to run the experiment and analyze the results. So we should start seeing results really quickly. I'm going to keep the video running on the gel chamber so that you can watch the dyes resolve into separate bands. And then when the experiment is done, we're going to compare the results to those in our database. And that's done at the end of, and we're going to do that at the end of the experiment. So what do we think of when we are designing a kit? So there's kind of 8 steps that I like to think of when we're designing a kit from scratch for teachers to use in their classroom. And the first thing I think for me that's most important is to research the science that is in the news and is in the curriculum. So we really want to pick stuff that's exciting to students to get them engaged and excited about the science that they're doing about in the classroom, and to show students how what they're learning is relevant to the real world and what's going on. So that is a big part of what we do is, we try to translate this science into an experience that students can have to connect with what's going on. For an experiment like this, an electrophoresis experiment, we would take the information from the research and think about, what we want the results to look like in our experiment. So in a dye electrophoresis experiment, we would try and figure out what the banding pattern that we would want would be. That could give us answers in a specific test. And so we do that using combinations of existing reagents or by creating new ones. And so, for example, our dye kits use many of the same dyes. So, you may see a red dye in this kit and a red dye in another kit that you might use, and likely they are the same red dye, because they're all negatively charged and they're all going to run through the gel like DNA. However, if we wanted to try a new dye, or if we wanted to try a positively charged dye, in our initial, development process, we would find that reagent, we would test it, we would see how it works with the other reagents to make sure that it would run the same way. So the next thing that we do is we would actually create the protocol. So we have the reagents, we want to teach electrophoresis, we need to come up with guidelines for the gel percentage, how long we run the gel, how many gels you can run at a time, what the length of the gel is. So here I'm running two 7 by 7 centimeter gels, but there are experiments where you need to run a full 14 centimeter gel to be able to view the results. And so, for our electrophoresis experiments, we do have a pretty standard protocol, but what we need to do is make sure that our experiment fits within these parameters. For new techniques, we're starting from scratch. So we are, doing the research and development to try and figure out how to get something to work best. Once we kind of have the new protocol in place, we test it. So not only does the researcher test the experiment themselves to make sure it's reproducible, we then pass off the reagents to another person in the lab to test. Because sometimes there are tricks that people do in the lab that they don't realize they're doing. And sometimes those little things can influence the way an experiment runs. And so we pass off the reagents to other people in the lab to make sure that they can get the same results that we do, meaning that the experiment is not only reproducible, but it's reproducible in other people's hands. So you could imagine, doing a transformation experiment or a PCR experiment. If somebody pipettes wrong, they might get different results. And we would want to figure out what that problem is and how we could fix that problem so the experiment is reproducible in a lot of different hands. And so, in developing a kit, we're not only talking about the science, but we do send a full literature with background, with questions, study questions, with some guidelines. And so that's actually a big part of what we do in creating the classroom experiment as well. So we review the current literature to write a background. We discuss research and discuss the technique. Because all of these things are important to the whole experience of doing an experiment. Because doing electrophoresis is great, but why are we doing it? What are we testing for, and how is this interesting? Once we have completed the literature, once we have tested the experiment, then we are going to send it to additional staff for review. And so that's important because, we are looking for typos, we're looking for mistakes, because, you always prepared your work before you hand it in, right? That's just good practice. Once a kit has been approved, it then heads to production where it is built. And so the experiment we're talking about today is a STEM kit. And so we do have a lot of experiments that we consider to be STEM experiments. And so what is STEM? STEM is a buzzword in science education for a long time now. But, we kind of feel like STEM lessons are those that are going to integrate science, technology, engineering, and mathematics into an interdisciplinary unit. STEM itself breaks down complex questions, challenges, or problems, or global issues into parts to discover its nature and relationships. This involves reading and research, not just the hard scientific literature, but sociology and the human implications behind a given topic. We then identify and understand content knowledge from STEM and other disciplines and use a line of reasoning to generate the necessary solutions to real-world problems and answers to complex questions. And so, in my mind, I really consider this good science. Biology itself does not exist in a vacuum. So, for example, I'm a molecular biologist, and when I did my PhD, I was working in a fly development lab. And so one thing we did was looked at fluorescent tagged proteins in fly eggs, which helped us learn about gene regulation. And so we used genetic engineering to make recombinant DNA molecules that were injected into fly eggs. Once the DNA was spliced into the fly genome, we used mathematics to calculate the probability that our fluorescent protein was going to be present in a specific egg. So, for example, if a parent fly has one copy of a fluorescent protein gene, 50% of the offspring are going to carry that gene. And then we used a very sensitive microscope to look at the fluorescent proteins within the fly eggs. And the results of this experiment helped me understand how genes are expressed into proteins in cells, which is important for human biology and health. And so misregulation of gene expression can cause diseases like cancer or osteoporosis. And so this is an example of STEM, of good experimentation where science, technology, engineering, and mathematics all depend on one another to work together. And so these are some of the things that we can show our students through hands-on experimentation in the lab. So let's talk about the molecules that we're separating here. We loaded our dyes with a mixture of brightly colored, we located our gels with a mixture of brightly colored dyes. And you can see, our gels have been running for about 10 minutes now. And we can already start to see them separating really well. Let me actually bring the camera down a little bit more so that you can see that. There is a little bit of heat generated, which does fog the lid a little bit. But you can see through it and see the dyes migrating. Especially when you run the gel at higher voltages, you do get that steaming because you are heating it up when you're, when you're running the current through the gel. And so let's talk about the dyes we use in experiments. So the structures of two commonly used food dyes are shown here. One which is yellow 5, which is tartrazine, and the other is blue 1, which is brilliant blue. And at first glance, they are sort of similar molecules. They're kind of the same size, and they all have aromatic rings. But when we look more carefully, because of their nature, the chemistry is actually different. We have different bonds, different functional groups, and all of these affect the charge of the molecules. So while both of these molecules are going to have an overall negative charge, but by virtue of the side green groups and bonds, tartrazine has a larger negative charge than brilliant blue. And because of these properties, we expect that they're going to migrate differently. And so we loaded our gels with the mixture of dyes. And, what does that mean when we actually look at the wells, when we look at the gel and we apply current? And so you would be correct in thinking that they are going to migrate differently, when moving through the gel because of their charge. And so the dye molecules have an overall net charge which influences their movement through the gel. Because molecules with different charges travel at different speeds, they become separated and form discrete bands within the gel. Some of the dyes are negatively charged, and all of the dyes in this experiment are going to be negatively charged, and that means they're going to move through the gel towards the positive electrode. Dyes with greater net charges will be more strongly drawn to the electrodes. So for example, if we think about those two charged dyes we looked at before, xylene cyanol and orange G, because of its larger size, we looked at brilliant blue. Sorry, I have made the wrong note here. But yes, so we looked at an orange dye and a blue dye, and what we'll see is that they had different charges, and so tartrazine, which had the larger negative charge, that one is going to move further through the gel, and it's going to move more quickly, again, based on its physical properties. Now, if we happen to have a positively charged dye, instead of traveling to the positive electrode, it's going to travel to the negative electrode. And so if you're used to seeing DNA gels, it looks like the sample is running in reverse. But basically, that is exactly what we would expect based on its chemical properties. And so for the kit we're testing today, which is S46, we wanted to create a kit that could be used to teach multiple topics. So we started with some current issues in biotechnology and in biology to create a detailed background. And so we thought of 4 scenarios that, our teachers, like to perform with their students. And we created background for all 4 of them. So the first scenario is a crime scene forensic analysis where scientists isolate DNA from a biological sample found at the scene of a crime. Students identify the criminal by comparing the crime scene DNA with suspect DNA. And if forensic science is something you're interested in, we have several live streams that focus primarily on forensic DNA analysis and the background behind it. Next, this kit comes with a scenario for cancer gene detection. And so your students are going to diagnose the state of the P53 gene in 4 separate tumor samples. And so we discussed cancer gene detection in a different live stream, which is available also on our YouTube channel. The third scenario is the detection of genetically modified organisms in commercial food products. And we do that using specific markers in a plant's DNA. And then the final scenario is a DNA paternity test where students learn about genetics and heredity by comparing the child's DNA sample to that from potential parents. And so using this kit, we can talk about that biology and biotechnology, but we can also introduce STEM techniques through an exploration of the ways that objects interact with one another. So specifically the interactions between the dyes, the Agarose matrix, and the electric field used in electrophoresis to separate these molecules by charge. We can also discuss cause and effect through the way that differently charged molecules separate in a gel, how different percentages of gel affect the migration of the dye fragments, and how the migration of the dyes are influenced by different electrical currents. And so I've actually tested some of that in a video on my YouTube channel, on the Edvotek YouTube channel. So you can check that out too, how the percent gel is going to affect the migration of samples through the gel. So when we create the literature, we wrote a common background about electrophoresis, but then we researched these 4 different topic areas to write supplemental material about each of the issues, and then gave some STEM exercises to do along with the experiment. And so those are that's kind of the way we conceptualize the background of the experiment, and how we write it and how we deliver it, thinking of what your students would be interested in learning from a particular hands-on experience. So after a kit is manufactured and it's on the shelf, our testing is not done. So we want to make sure that an experiment lasts through storage at our facility, through shipping, and then storage in your lab. So when we do our QC, I hit my light. When we do our QC here, we know the results look great on a specific day, but we want to know how it's going to look over time. And so we create simulations of shipping, and we store our kits at different temperatures, and then we test them at regular intervals to determine their shelf life. So one example is shipping stability. During different seasons, the kits may undergo different temperature changes while they're being shipped. So for example, in the northern hemisphere, during the winter, you can get some freezing temperatures, or if we're sending something, to a different hemisphere, you might have a different season. And so, for example, in the summers, it's too hot to ship restriction enzymes. We know that they're going to have problems, and we need to ship them on ice, unless they're in our dryzyme format. In the winter, our C. elegans, or the cells for our cell culture experiments, can freeze if they're left on a shipping truck for too long. Plus, we're located in DC, and we have customers all over the world, so kits can end up in transit for long periods of time. And so we set up simulations in our lab to approximate what would happen to a kit during time spent in transit. And that can include keeping a kit on a rocking platform, putting it at different temperatures, and then we would count over days and take the samples to make sure that they still work right and they still last. We also want to determine how long a kit will last once it's built, so we perform long-term stability testing. Again, we're going to place the kits at different temperatures, and then we're going to test them at regular intervals. And then we can give you the guidelines for storage and use in your classroom. And of course, if you have any questions about the storage conditions, let us know. And so, again, after taking our samples and doing the tests, what we have determined is that for our quick strips, you want to store them in the refrigerator. So we label it on the packaging. We're going to label it on this insert that comes with the samples. And then we also tell you when to use it. So you want to use the kits within 9 months of receiving it. That doesn't mean that it won't work outside that time period, but this is kind of like for best results, use within. And so there are some reagents that will go off in a short period of time, and we give that to you as a short period of time. If it's a school year, we're going to tell you what's good for a school year. Again, if you have questions, definitely ask us, and we can help you determine whether or not your kit is usable. But, even under the best of circumstances, sometimes there are problems, If a problem is identified in our initial QC, we repeat the experiment, testing each component to see whether it's working right. if a teacher gets in touch with us after having a problem with an experiment, we ask for results pictures and for the lot number. And so these are really useful for two reasons. The picture is really useful because sometimes we can look at a gel or look at the results from an transformation experiment and see what happened. Especially with electrophoresis, if you see nothing on a gel, maybe you reverse the polarity. If the samples don't run through the gel, right, maybe you used water instead of buffer or something was diluted incorrectly. If you're doing PCR, especially some of our human testing kits and you don't get any results, maybe you used the wrong buffer or you used Gatorade to extract the DNA. And we know that doesn't work with our kits. So giving us the result pictures and talking us through what you did, that helps us to start identifying places where something might have gone wrong. When you give us the lot number, what we can do, so again, here is our lot number for our quick strips. When you give us this lot number, what we can do is find some of the strips from our long-term stability testing and go test them in our lab and make sure and see what happens. So if we get the same results that you do, something has gone off in that batch and we need to test and pull that off of our shelves and not ship it out anymore. And so this is important data because it allows us to figure out what went wrong with which reagents and how we can fix that problem. And so if the problem is not obvious from the pictures and it's not obvious from, we then perform the experiment. And through these means, we can then identify what happened with the kit, what went wrong, and how we can fix it for next time. So we are almost done with our electrophoresis experiment with our QC. I think we've got another 2 minutes in our 20-minute runtime. But I want to just recap what we've done so far. So before the workshop, I cast the Agarose gels. We placed the gel into the gel chamber. I covered it with the electrophoresis buffer, and then I loaded the samples using an adjustable volume micropipette. We placed the lid on the safety chamber and then turned on the power supply to generate the current necessary for electrophoresis. While we were talking about quality control, the dye samples were separating into their different bands based on their charge, and now I think we are ready to take a look at our results. So let me move our camera back up again. I'm going to turn the power off. So one thing to note is that, when we designed this chamber, we wanted to make sure your students couldn't stick their fingers in here while it was running so they didn't get a jolt. So the best thing to do is to turn off the current before you remove the lid. But since the electrodes, here, let me move up a little bit. Since the electrodes are built into the lid, we can't have the current running through the chamber when the lid is removed. And so that's important because it basically means that, you are not going to get shocked. All right, so I'm going to take this gel out and I am going to place it on our True Blue 2, which is our transilluminator. And so one thing I like about this transilluminator, so generally we use these transilluminators. This is a blue and white light transilluminator, and so you might use this for your cyber-safe stained gels. But one of the, I think, underutilized features is this white light visualization system, which is perfect for dye electrophoresis gels. And so what it does is it's going to illuminate the samples from below and really allow us to see the good contrast between the samples. So let me turn that on. Make sure to turn it the correct way. And so you should all be able, let me center this up a little better. All right, so you should all be able to see those samples. And so what you'll see is that each dye sample, so we have sample 1, 2, 3, 4, 5, 6, migrated through the gel in a different way. These dyes are all negatively charged, though they all have different net charges, which is going to influence how far they move away from the wells. And so, for example, what we can see is that the orange dye in lane 1, migrated really far through the gel, further than this blue or this purple, and so it probably has the largest charge. So in lane 1, we loaded a combination dyes that separated into distinct bands when the current was applied. The fact that they were mixed when we loaded them doesn't affect how they run through the gel. That's what electrophoresis does. It separates things based on their physical properties. And so, we can see that each dye is going to migrate and each lane migrates in a different pattern. So if we were performing the experiment, we would analyze the patterns according to the scenario which we chose. We could discuss the implications of the different banding patterns and what they mean in terms of genetics and heredity. And so for QC, what we are asking is, does the banding pattern match? And what I would say is, in general, yes, the banding pattern here does match the picture, but we do have a little bit of a difference in lane A from what we were expecting with the 4 separate bands. So what I would probably do is contact our production department. I would talk and then we would discuss the appropriate next steps to take. So we have come to the end of our experiment. We've covered a lot of information over the course of this experiment. We've learned how Agarose gel electrophoresis can rapidly separate molecules in discrete bands based on their size, shape, and charge. And so while fairly simple, we just did this experiment together in my basement. It is very powerful when it comes to rapidly separating different molecules from one another. Edvotek designs our experiments around real-world science, so even with simple experiments, we research the science to make the kits relevant, and we perform the experiment time and time again to ensure that the experiments are reproducible and that they'll work in your hands. And through our quality control protocols, we make sure that our experiments provide reproducible results so that when you do science with Edvotek, it works. And the one thing I do want to emphasize is, if you have a problem, don't hesitate to contact us. Give us a call, email us, and send us pictures and the lot number so that we can help make things right. But we want to hear from you when things look good too. So definitely send us pictures on Twitter when your experiments work. If you are on Instagram, tag us in, because we love to see you doing science and we love to see how it works for you. And so through our quality control protocols, we make sure that our experiments provide reproducible results so that when you do science with Edvotek, it works, so that you can send us those good result pictures. And so we are going to be posting the presentation and the slides in the next few days to our website, which is www.edvotek.com. If you want us to email you when they're available, please again fill out the form that Maria has posted, and we will send that to you, same as if you want a professional development certificate. So, thanks for your time. If there are any questions, you should ask them now. If you need us for anything, don't hesitate to reach out to us at info@edvotek.com or through one of our social media channels. We really want to help you get biotechnology into your classroom any way possible. And so you can call us. We can run workshops for you via Zoom, to help your teachers get comfortable with biotechnology. So definitely just let us know. And so with that, thanks again. Have a great day, and hopefully we'll see you at another biotechnology live stream. Have a good one. Bye.